Okay, I think we can restart. And we start with a very attractive topic now, that is artificial intelligence and a bit understanding dementia in some way. So we have the privilege to have with us the Professor Jing-Dong Jackie Han. I hope I didn't destroy your name. Who is Professor Center of the Center for Quantitative Biology at the Peking Tsinghua Center for Life Sciences, Academy for Advanced Interdisciplinary Studies at the Peking University. So we are really very curious to listen to your lecture. Please.
First, I'd like to thank Professor Comi for the introduction and also Professor Shi. It is my great pleasure to present here. Among all the physicians, I am the only one who's not working on in clinics. I'm a basic scientist, a computational biologist. Okay, where is the other stronger laser pointer? I thought there's one. Okay, I'll just. So what I'm going to talk today is AI and aging. Everybody here is probably very familiar with aging, either from your family members or anybody on the street, right? Aging affects every aspect of our human body during the process. For example, you can see the appearance on the face. We already talked so much about the aging brain. On the left is the young, on the right is the old brain, if you can see here. And also in the bone, you can also see the bone become more porous and the muscle become fatty. In fact, not only the appearance of aging is affecting us at the tissue level, but also at the molecular and the cellular level. So on the right is the hallmarks of aging. This is already an old slide. Last year, there's an updated hallmark, now including 12 hallmarks. Mainly is the genome instability, telomere attrition, and cell-cell communication defects, etc. But in the last updated version, they added systemic inflammation as a major driver for aging. In this old version, there's no inflammation here. Aging, although it's very complex, it's quantifiable. In the past, actually, I think it's already a decade since the first aging clock was published. Aging has been measured at many different levels, from proteome, transcriptome, methylome, any omics data, you name it. You can use this omics data to build an aging clock and estimate the aging progress. Essentially, an aging clock is built on large cohort of natural aging population. For example, here, this is the first molecular clock of aging. It's a DNA methylation clock measured, built on 656 individuals. And you can see, based on this CPG arrays, I think they use around a few hundreds of CPG sites. They can build a linear clock, linear model to predict the chronological age of anybody's age. When you see this plot, predicted versus actual, or oops, I gave out the. Okay, here's the laser. You can see this predicted age and the chronological age fairly align fairly well on this line that indicate the prediction is very good for the majority of people. However, there are some people appear to have higher predicted age than their actual age. And these are named fast ager. And on the other hand, there are other people who seem to have very low predicted age than their much lower predicted age than their actual age. Those are the slow agers. Basically, this indicate, here's the picture, for two individuals of exactly the same chronological age, they can have very different biological age, at least DNA methylation age. Then we asked, you're all laughing because you already can see who's older, who's younger, right? Does anybody think this one is older? Yeah, you all agree it's the opposite, right? So we thought if you can already tell a person's biological age on the face, why don't we just look at the biological age by the face? That's the first experiment we did. We collected 332 individuals from Beijing CDC. And here shows the average face of each decade, average of 30 individuals of each decade. Okay. From 20s, 30s, 40s, 50s, and 60s. And you can already, by your eye, look at the features of aging on the face, right? For example, what do you see? The number of wrinkles increase, right? And also the eye corner droops. But some subtle changes you probably didn't notice unless you build a clock model. So what we did was using this partial linear square regression model, we built first a linear model because this linear model can be well visualized on the average face, which is in the middle. This is the 3D face, right? And this changes on this average face can be visualized by the first two components of this clock. Roughly, that explains around 70% of aging-related changes on the 3D face. And you can see what you just noticed is already captured here, but somewhat exaggerated because we're stretching it to the two extremes. This roughly corresponds to 25 to 30 years younger, and this is 25 years older. The eye corner droop with aging, but also there's something that you didn't notice but exaggerated by our model is the forehead size decrease with age. And I think this really has something with the brain volume also decrease with age. On this, this is another way to illustrate the changes. First is that on this x-axis, that is the horizontal direction. Going to this direction means it's going to the left. Because the image is centered, going to the right is showing on the right is showing as the blue. So this basically indicates the forehead size decrease this way, and the lower face increase on the horizontal direction, and on the y-axis there's not so much feature, except this region, the bridge of the nose seems to move upward. On the z-axis, the depth actually gives the most information. If we take out the z-axis, the accuracy of the model decreases a lot. That's because many of the aging-related changes are written on the z-axis. For example, first it's the forehead decrease in size, the depth or the height of the forehead decrease, and also the eye area protrude with age. Here with this linear model, based on 300 people, we can already achieve an error of only 6 years. Using this 6 years as cutoff for defining the fast ager and the slow ager, we can see the outliers for this model do show some aging-related changes. For example, in this, these are the people of the same chronological age group. The fast ager or the upper outliers do look older, right? And the lower outliers or the younger outliers do look younger. This is similar for different age groups for females and males as well. Not only aging is written on the face, but other molecular markers can be also detected on the face. For example, cholesterol level shows somewhat similar pattern with aging. Both the total cholesterol level, low density cholesterol, LDL, and high density cholesterol is showing this opposite pattern as the two upper lines. And albumin is very unique. In particular, albumin in men is very strongly associated with the forehead size. And more recently, we have now expanded this analysis to a much larger cohort with 5,000 individuals. Now we can use AI instead of a linear model to build an even more accurate clock. With this clock, the error reduced to 2.8 years to the chronological age. And in this cohort, we also introduced another clock, the so-called perceived age. Perceived age is what other people look at your face and determine what is your age. So this is basically artificially determined age by people judges, human judges. And you can see the perceived age has a much larger error than our CNN AI clock. It's 4.5. But it's very similar to the linear clock in the same built from the same cohort. However, if we train another AI model to learn how other people perceive your age, so this is called the CNN perceived age. After we have this, we no longer need judges to judge everybody's age. Instead, we have this AI to pretend to be human judges. But this AI model actually corrected some of the human errors, reducing its error to chronological age to 4.1 instead of its input of input training data of 4.5. And surprisingly, it is this CNN perceived age most significantly associated with all the different lifestyles and different health parameters that indicate, well, what's showing here is the age diff, or the difference between predicted minus actual age. A positive age diff means accelerated aging. And you can see here this perceived age, CNN perceived age associated strongly with many health parameters, in particular BMI, weight, all of those bad parameters. And also, it associates with many bad lifestyles, such as smoking and snoring. And at the molecular level, we're also interested in what is reflecting when you look at somebody is showing accelerated aging. So to answer that, we also tested the blood transcriptome of the 300 individuals from Beijing cohort. So we not only, for this cohort, we not only have the facial clock age diff, but also this blood age diff. For any of this clock, the most significant association with the transcriptome data is actually infection and inflammation. Inflammation is particularly shown as here. We found that monocyte count in the blood is significantly associated with both the accelerated facial aging and accelerated blood aging. And if we directly visualize the monocyte count on the face, it shows like in this corner, mostly it's a protrusion of the nose and the mouth area, indicating if there's more inflammation, this center of the face protrude more. And at the bottom, it shows how this goes with the 3D contour plot. Essentially, with more monocyte, the face looks older, right? Since we have not only the transcriptome data, we also have lifestyle data, very quantitative lifestyle data. That's why we can build a so-called causal inference model to infer what molecules in the blood may mediate the lifestyle impact on the face and also on this RNA clock, blood RNA clock. What we found, of course, as expected, smoking and drunkenness accelerate facial aging the most, and both of them are mediated through inflammatory factors in the blood. Many of them are cytokines or cytokine regulating transcription factors. On the other hand, some good lifestyles, such as yogurt consumption and even coffee consumption, modify the slow down facial aging, mostly through epigenetic factors. One thing I want to mention here is that in the foreground, what we showed are all those coding genes. In fact, in the background in this network, these are the non-coding genes. You can probably appreciate that the non-coding genes are many more than the coding genes as mediators. So what are those non-coding genes? We further analyzed the non-coding genes associated with aging, found that compared to aging non-related link RNAs, long non-coding RNAs, aging-related link RNAs are more conserved throughout evolution. And surprisingly or reassuringly, we found that the top enriched motif on this link RNAs are actually controlled by the #1 inflammatory master regulator, the NF-kappa B transcription factor. Then through a CRISPR library screen, we also found many of those link RNAs also feedback regulate NF-kappa B, forming a vicious inflammatory cycle or positive feedback. Once it's set on, it's very difficult to set off. Among all these link RNAs, we also found a very interesting, very long and very abundant link RNA, which is called KCNQ1OT1. It's 90 kb. What's interesting about it is that it specifically targets transposons. Those are the mobile elements in our body. In fact, our body, our genome, are mostly compressed of transposons instead of coding genes. For example, the Lu elements, there are 1 million copies in our body. And for the L1 elements, there are 500,000. If they are activated, it will jump all over the genome and basically damage our DNA. What we found was that this K-link specifically recognized those L1 and Lu that can still be active. Those are the young, very evolutionarily young L1 and Lu, and keep them in the repressed state by recruiting repression machinery. Well, to summarize all those lincRNA functions, I recently wrote a review. I actually very like the picture I drew. Is the damaged nucleus. You can see nucleus like our human face become wrinkled and basically a mess after the cells goes into aging or senescence. Many lincRNAs are associated with all these structure formation complexes. So here I just propose that maybe in the future, coupling the image of cells using AI approach and also perturbation of the lincRNAs, we can identify what lincRNAs is controlling what factor, what structure of the nucleus. And in addition to our facial clocks, RNA clocks, we're now also moving on to single cell clocks. So here, in collaboration with Dongfang Hospital in Shanghai, we collected 17 individuals, and also together with another 11 samples from Guangdong Medical Hospital, we built a single cell clock. For this single cell RNA clock, we can see that the cell type contribute the most to our aging clock is the cytotoxic CD8 cytotoxic T cell, which increase dramatically with age. And also the two naive CD4 cells and naive CD8 cells decrease with age. We applied our clock on supercentenarians and also on COVID-19 and lupus patients. We can see that supercentenarians usually show a very delayed aging, whereas the COVID-19 patients, the severe COVID-19 patients also show accelerated aging in the single cell blood clock. And lupus actually show a very dramatic increase in their single cell clock. If they're managed, if they're properly treated, this accelerated aging can be brought down. However, during this analysis, we found that even though we have this clock, if the next patient or the next normal person comes in and wants to measure their biological clock with single cell RNA-seq, the problem is how can we integrate those two data? It's very difficult to merge them together and calling the same cell type exactly the same name. That's why we recently built another AI model. It's called Transformer for One-Stop Interpretable Cell Type Annotation, or TOSICA. Now this tool is very, very popular in GitHub. Almost every week we get several requests of how to use it, and I got many invitations to talk about this too. But if somebody's analyzing the single cell data, you're welcome to try it out. In the remaining few minutes, since this is a brain aging symposium, I'm going to talk about a little bit about the brain aging. especially brain disease. We recently collaborated with Jing-Ye's group in Beijing Hospital, where their group collected around 200 stroke patients, and we matched this 200 with 3 times more control patients from age and sex matched control patients from Beijing Hospital, also the emergency room. One X is from Beijing Hospital, and the other 2 folds are from Jing-Dong Hospital and also Fuqing cohort. Because we have to have age and sex matched controls. It's very difficult to find all of them. And surprisingly, by just looking at the face, we can already achieve AUC of 0.91 for our cross-validation. And even in a totally independent cohort, the AUC is 0.86, I think. The accuracy is also about 0.8 something. In fact, this is already higher than the current CT or MRI. And I'm going to show you a more detailed comparison later. So what are we looking at? Because our predictor is now we're applying for a patent of our predictor, I can only show you some rough attention on this AI model. Mostly on the 3D phase, the stroke patients shows a higher nose point compared to all other points, indicating that there's a global drooping of the face or paralyzation of the face. And surprisingly, we can also see our predictor is not only yes or no prediction, it's very quantitative. Here shows just the cutoff for our predicted stroke versus the predicted non-stroke. They are also associated with many blood indicators known to be associated with stroke. For example, the blood clotting factors such as, where are they? I can't see, FIB is known to be a predictor, not a predictor, but an associated factor with stroke events. However, we also identified several unknown associated factors. For example, the serum phosphorus is significantly anti-correlated with the stroke probability. Another interesting observation we had was in this region cohort, the control cohort, who are the 200 also patients also visited ER, but for other disease, diagnosed not to be stroke. We found the prediction score is very different from the general population, which the general population is the blue, whereas the region cohort, the region control cohort is the yellow. You can see it has a very large overlap with the region positive cohort. Using this probability of 0.45 as to determine who's going to be who's going to be negative, who's going to be positive, we can see that around at least 1/3 of the control also have a very high probability of stroke. Then we asked if these people actually have the tendency of getting stroke. Indeed, their blood parameters also show higher association with stroke risk factors, such as these two clotting factors. And our collaborators also recalled all these patients and found indeed in this false positive predictions, those are the ones we predicted as stroke, but the people, they are not diagnosed as stroke. They have a much higher odds ratio to be stroke. 5 out of the 34 people indeed developed a stroke in the last 2 years, even though they were not diagnosed. Whereas in this prediction, true negative, there's only 1 incidence. So that indicate our predictor is, although it's derived from cross-sectional analysis, it has the capability to predict future event. So we are looking forward to look at even more longitudinal data to confirm whether the cross-sectional model and the longitudinal model shares some common features. With that, I'd like to stop here by first acknowledging all the people who did work, especially our collaborators. I have many clinical collaborators. The Jing-Dong cohort was collected by Yongzhou from Jiao Tong Hospital, Jiao Tong University Hospital. And the stroke was a collaboration with Beijing Hospital. And the single-cell RNA clock was a collaboration with Dongfang Hospital. Thank you.
Thank you very much for this so interesting talk that also, by the way, touch everyone here. So, which is now open to question. Maybe I take the privilege before to start.
Brief question, brief answers.
Yeah.
Thank you, Professor Han. I have one question. Is it possible that we can decompose the AI prediction into such as the feature vectors, which will reveal the true problem as the muscle chain on the faces, which eventually causes the change of the facial pattern? Thank you.
Yes, we can now do interpretable models to, especially with transformer models. We can, it can be more interpretable. In fact, even with what I presented are mostly CNN models. There are also ways to identify the area of very important weight to the model. As I showed you, like the stroke, at least on this association, is the nose versus the rest of the face. In fact, there are also other features that, because we're patenting, I did not talk about. Yes, we can see very precise area of attention. But for muscle, I don't think we can actually see the muscle fibers. It's not that resolution.
Yeah, have you, I mean, here there is a lot of interest trying to understand which are the variables that influence the evolution of neurodegenerative disease, for example. Have you had any tentative to try to predict the speed of evolution of dementia in Alzheimer or of multiple sclerosis? Because that could be extremely interesting. No doubt that the aging factors have an important role in the process of neurodegeneration, and on the top of that you have the effect of inflammation. So have you already such an approach?
We are starting to collaborate with hospitals to study Parkinson and also depression with various different grades. But so far I haven't, I really want to study Alzheimer's, but I haven't found a collaborator yet.
We could be. I mean, my question would be that we are in close proximity in
Yeah.
Beijing, while you collaborate always in Shanghai. So,
Oh, because I worked in Shanghai for 8 years before
right.
I moved to Beijing.
so we can realign for collaboration. Probably, we can talk more about that because the proximity is important. Maybe my question would be that. So because we use facial recognition for getting on board a plane, going to work, because we are aging. I mean, how many years you think that we should adjust the matrix so that the recognition will be accurate?
Oh, actually the recognition is very accurate
Even
across
when
many different decades. In fact, we have tried different ways to deform the face. The recognition is still very accurate because it relies on the relative position of, and then that signature of the relative position is very individualized.
So, what is the rationale for you to select the biomarkers in stroke patients and in correlation with facial change, and you consider high throughput, and why do you select those?
High throughput. So far, high throughput, we cannot do single cell RNA-seq for high throughput. I don't have that much money. But yeah, we do, we're now working with a lot on tongue microbiota, which is much cheaper to profile, and also everybody is willing to just rub their tongue and give you the sample.
Right.
Yeah. But you're welcome if you have a technology to do high profile cheaply.
Yeah.
Okay. Very good lecture. For me, I see you say the AI and the face of some aging. For me, I often use say how to research the spirit or the self consciousness or self orientation for some spirits. Maybe for some biological basis, maybe can do some research. 就是关心的，我们都要关心面部的表情。
So basically, we are focusing on the facial aging, but for like the spirit aging, self-awareness in terms of biological foundation. Any plans?
Traditional Chinese medicine doctors to understand the personality. In fact, from the face, the personality can be. I cannot see very accurately, but at least by an AUC about 0.7, we can predict the personality.
Right, if you don't mind, I use Chinese.
So is that a prediction or identification? Because the training set is a kind of known material, for example, to feed the AI with the stroke patient data.
Actually, I have another slide to address this. But I already showed you this prediction in the control cohort. Those people, we determined those are essentially prediction, even though our classifier was trained to predict the class. So initially, our model is a classifier, but it has capability to predict future events in at least the past 2 years. Yeah. Yeah, in the past 2 years, we have 34 patients, which we found they have a very high probability of stroke by our AI, and 5 of them already developed a stroke. Whereas in a low probability group, only 1. So that odds ratio is roughly 5. That's why we think it really has a prediction power. And we're excited to just hopefully it can be applied every day to everyday life to monitor everyone.
Thank you for the talk. There was a paper recently about the relationship between the face characteristic in Han Chinese and the genome, especially with the single nucleotide mutation as SNP. So because you have clear aging differences just by the different parts of the face, do you think if you take this look into this database, they found about, I think, 100 SNPs or something which are responsible for the different phases, could be helped to find out some markers for aging?
Yeah, we actually collaborated with that group. Actually, Sijia was from my old institute in Shanghai. So when they mapped the SNP and we mapped the aging rate, we did an association. There's really almost no signal, except one weak signal in telomere. But telomere, I don't know why it's the sort of opposite to our expectation. So really the aging rate difference across different individuals are generally not genetically determined. People have studied the twins and found that aging rate difference across 2 different individuals are 90%, at least 85% epigenetic, only lower than 15% genetic.
Other questions?
Thank you.
I'm not a big fan of monitoring everybody with this technique. 我说我不是喜欢
I'm not the person to advocate the monitoring.
by abused, whatever, by whatever
By
organization.
insurance company, maybe. Yeah, it could be used by insurance company, and I hope insurance company can buy our technique.
Well, even in politics, perhaps. Okay. I can stop here. I can stop. Okay, thank you so much, really. And I would like now to ask our co-chair to take the podium and just to address some consideration about inflammation and neurodegeneration. And I think you are really a deep expert in this area, so we'll be happy to hear you.
All right. 我试图想就是。
I like to speak in 2 languages. So my presentation is not very long.
Lingual and so that everybody can understand without a translator. And I used to speak some Swedish, but I lost it since returning to China. Well, this is an attempt to have a bridge between the morning session and afternoon session. Well, in the morning session, we talk about neuroinflammation, MS and MO, and its measurement. In the afternoon, we talk so much about neurodegeneration. Well, so 我试图想就是上午下午的课题能够。
So we like to link the morning session and afternoon session.
Task to summarize neurodegeneration across spectrum of disease. I'm going to skip this. This is an attempt to compare the kinetic and the magnitude of inflammation across the spectrum of neurological conditions as manifested by multiple sclerosis, acute brain injury, and degeneration. So this is by no means accurate, but it's illustrative. 这个不是试图想确切的。
So it's not to accurately to display the degree of severity or the dynamics, but this is just a kind of conceptual illustration.
Cells homing to the brain, and over the time, the magnitude of inflammation will tune down, then the neurodegeneration process will kick in. So this is what we currently hold in the field of MS. On the other hand, in case of a stroke, either ischemic or hemorrhagic, or traumatic brain injury, or in the case of status epilepsy, there is a pronounced inflammation that's caused by the cell death product. So, however, we don't really know how long the inflammation persists, or if they disseminate across the brain over the time. 就是在这种急性脑损伤的时候。
So upon TBI or the other acute injury, there will be a lot of cell death, but how long it will last and will that spread across the brain, we don't know. And in the degenerative diseases, so Dr. Chen just mentioned there is a gradual cell
reduce increase of inflammation. So I think it's useful to consider this magnitude in these different conditions, so that whether we should have any therapeutic application. This is just appear in Cell, this review, again attempting to summarize the common elements that occurs in Parkinson's disease, Alzheimer's, and MS. So you can appreciate that inflammation does exist in all these spectrum of condition. Then the neuronal cell death, pathological protein aggregation, and synaptic and nuclear network defect, and DNA and RNA defect. So altered energy homeostasis, cytoskeletal abnormality. So there are really no specific elements that are specific for each condition. They really share these pathological condition. Well, not everybody sitting here is a neurologist. So let's talk about multiple sclerosis. And Jacqueline Dupree is a very gifted cellist from Sweden, from Britain. She suffered from MS in the age of 30, and she died at the age of 42. So I would love to have Dr. Chen's approach to study her age, lifespan in her approach. But you can see during the age of the 87, we don't really have any disease modification therapy. So nowadays, if she is cared by Giancarlo or myself, she would have a very different fate. Not only she may be able to continue to play, but so she should probably live a very, very relatively normal life with normal lifespan. So this is saying that how much progress has been made over the past 40 years also in terms of the ability that how we treat this patient. So this is to consider that why the disease progress, and there are several schools of thinking that one is immune dependent. We can see that within the brain you have microglial astrocytes orchestrate inflammation that's still ongoing even in the later phase of disease. They're not disappearing completely. On the other hand, you also have the influx of the immune cell. So they coordinate to establish this so-called compartmentalized inflammation that Dr. Comi mentioned in the morning. We have to note that the spectrum of immune driving force is very different than the case of CIS or early phase of MS. In that, innate immune cells are the driver, and also brain intrinsic cells probably play a more prominent role in driving progression of disease. The so-called immune independent mechanism are illustrated here. You can see energy deficiency, mitochondrial injury, demyelination in relation to ion accumulation, oxidative stress, calcium influx. So none of these are specific for degenerative process in MS, as we alluded in the prior slides. I'm not going to take you through this very detailed that how many of those process will induce neurodegeneration in MS. But this paper really was to, a couple of years ago, still stands very well now, showing that all these pathological elements are similar across MS and Alzheimer's disease. So we think that first, neurodegeneration does cause inflammation as time goes by because they produce so-called damage-associated molecular pattern. They aggravate microglia. They also attracts peripheral immune cells into the brain. On the other hand, the continuous neuroinflammation are detrimental in many ways to neuronal cell, axon, and many components of the myelin. So they are interconnected, and this is what we think currently. Professor Chen talked about the methodology to monitor neuroinflammation or degeneration. And for those, again, not familiar with Simoa, this is a technique that are able to detect minute amount of protein that are released from the brain to the periphery. So you can see the comparison of concentration between those within the brain and in the periphery. So the technique allows us to really picking up the trace of brain damage in the periphery without doing a lumbar puncture. I think this is useful for screening, for large population monitoring, and we started this technique a couple of years ago. Now we have a newer generation of Simoa in Beijing. This is one in Tianjin General Hospital. So I think it's very promising to collaborate with the biochemist to develop individualized, newly discovered marker to enrich the panel so that we can really monitor neurodegeneration. This is illustrating the usefulness of neurofilament light, tau beta amyloid across this neurodegenerative disease, while including MS and stroke. we don't need specificity, right? Because in the case of NFL, it's an element of axon. Whenever we detect that, we know it's a destruction of axon will leading to the release of this protein into the CSF or peripheral blood. So specificity is not an issue. Sensitivity is most important. I think the past few years really revolutionized our way to monitor disease, and hopefully in MS it will be a surrogate marker for the future trials. What about therapy? Do we have anything enhanced to slow the neurodegeneration? Here's a paper written by Heather Yang and Wei Yang, just the start of COVID, and Heather is a first-year neurologist, a daughter of Wei Yang, and so she working with his father to summarize the criteria for progressive MS. So I just want to illustrate a few points. So if you want to slow the neurodegeneration, the medication has to be able to penetrate the brain, and which has been alluded by several other speakers as well. So it has to have the ability to neutralize the focal inflammation within the brain. And it also should probably have an ability to tame microglia, because they are really the main driving force for the focalized inflammation. On the other hand, if it can acting on mitochondria, oligodendrocyte, it would be great. But I don't think single medication would fit all these needs. Well, having this in mind, we did a study a couple years ago by Zhiguo. He pulled the sample. This is a patient suffer from intracerebral hemorrhage. You know, in case of large hemorrhage, some of them has to go to OR to decompress the brain, because otherwise it was herniation. So by doing so, we got the tissue that's surrounding the hematoma. So the RNA sequencing allows us to pull this fomin peptide receptor out, and we think that's a major driving force to attracting neutrophil, and probably we are IL-23 as well. We also create a small molecule that antagonize this receptor. So we think that's one of the potentiating effects in inducing brain inflammation. Here we try to illustrate the so-called compartment inflammation. Well, this molecule mostly will acting on glial cell within the brain here, but it is also acting on the peripheral monocytes. So we thus think this approach may fit for several of the criteria described by Heather. Having this in mind, we try to visualize. Okay, here we go. So we try to visualize if there is a expression of fomin peptide receptor in MS autopsy brain slice. So you can see in comparison to the control, there is a higher expression of this receptor, and it's predominantly localized on microglial cell, to some extent on astrocytes. So it does leads us to the assumption that it may somehow curb brain inflammation within the brain. And we also did correlation of expression of its ligand in the peripheral blood. So the cohort is small, but we did see the tendency of correlation. Then we move on to knock out FPR1. And here is one of the models that mimicking acute attack of MS in a C57 black mass. So when we genetically eliminate this gene, the neurological deficit is attenuated. And also, when we adopt this small molecule blocking this pathway, we basically reproduce what we see in the knockout situation. And the myelin quantification, again, reflects the neurological condition. We have sophisticated neuroimaging tools in our institution, like 11 Tesla MRI, PET-CT, to see live animals, the demyelination and the inflammation process, and all these pointing toward it has a beneficial role in curbing inflammation and preserving the myelin. So I said EAE in black 6 mice, mostly mimicking an acute attack. So then we turn to another model in NOD mice where it's thus has a secondary progressive form of MS. So again, we see similar situation as what we did in a C57 black model. So thus this molecule has any way to reduce axonal dysfunction, which is a hallmark of neurodegeneration in multiple sclerosis. So this is what Yulin did in the knockout, as well as in B6 mice, as well in NOD mouse. So in this case, he sees synaptic integrity pretty much altered by knocking out. When we apply T0008, it's reverse effect. So it does have suggests us it has some role in reducing neurodegeneration. The other experiments she did was looking at the glial cell phenotype. She also did a single cell. I don't have data from her to show you. So it does seems to alter the inflammatory property as illustrated by the reduction of release of TNF-alpha, I1 beta, and ROS. She actually visualized ROS in a whole animal imaging experiment. So I'm going to switch gear, talking about inflammation in Alzheimer's condition, and Professor Chen and others really has covered the story very well. And again, for those who are not neurologists, and you may see these two movies, that I will talk about this movie that was released in US 2014. So Alice is a linguistic professor from Columbia. She went to UCLA to give a seminar. And during the seminar, she couldn't find word. So this is the first sign of Alzheimer's. And when she returned to New York, the condition quickly deteriorates. She couldn't hold her faculty position, and in the end, she wanted to commit suicide. She had a plan recorded in her computer, but she couldn't execute the suicidal process. So it's very, very touching, and so it's telling us that it's not an individual condition. It has a huge emotional, family, and social impact. And it is very moving. It's just the magnitude of its impact is just enormous. And again, the inflammation has its two phases, and I'm not going to through this. Professor Chen has shown these slides in his presentation. I want to make an additional point that in addition to aducanumab, lecanemab, see how much efforts has been devoted to anti-inflammatory therapy in this condition. So I think I can see the number is 17 therapy already under the phase 2 stage. So during the break, I talked to Professor Chen, probably antagonizing B cell or with tocilizumab or the other B cell could really be one of the addition to this already very vigorous investigation. We know this trial AL001 and the discovery were initially made in Washington University by my colleague and friend, Dr. Malano. He has a paper in Cell illustrating the homeostasis of glial cell. It's very important in directing the inflammation within the brain post Alzheimer. So then it is predominantly mediated by TREM2. So this trial is designed to blocking TREM2 so that to modulate inflammation to see the outcome. And I heard that the recruitment of this trial will be finished next spring. So we look forward for the outcome. And this paper just appeared a couple of weeks ago. Mestinib is a oral tyrosine kinase inhibitor that inhibit mast cells, microglia, and macrophage. So this trial, well, the number is not very big. So it does showing that administration of this molecule has some effects to slow the deterioration of cognitive function. And so we see that anti-inflammatory properties on the horizon, and we see already some promising tendency. And my fellow checked all the registered trials, and many of them are very familiar to our neuroimmunologists, like daratumumab is anti-CD33 molecule, as Chow talked about, is depleting the plasma cell. And this list goes on. I'm going to skip the Parkinson and inflammation because it's the death of the dopamine producing neuron, again causing inflammation similar to those in AD. So again, this is an anti-inflammation therapy that's going on in Parkinson. So going back to the situation of Alzheimer, with the lecanemab, whenever it's reduced pathology, it will reduce cell death. It will exacerbate inflammation. So if this is the case, it's probably going to offset any benefits that for cognition because of inflammatory process. So the point I'm going to make is that modulation of inflammation could be an adjunctive therapy for the future when we are trying to reduce the pathology. Well, simultaneously, the inflammation is going to boost it. So it has to be a big consideration or a risk for these monoclonal antibodies. We will wait and have to see more patients exposed to this therapy and how it goes. Well, this is my last slide. I don't think I provide any answer to this question, the similarity and differences of neurodegeneration across this condition. And we do think that inflammation causes degeneration or vice versa. And we have some tools to monitor the degeneration, but those tools are far from ideal. And we should be able to using proteomic or genomic approach in conjunction with AI probably, so that we can have a better matrix to monitor degeneration. And we do see some hope that if we are able to modulate neuroinflammation as what we did in MS, maybe this neurodegeneration disease will benefit from this approach. I think for the moment, we only be able to see this portion of Shanghai, and underneath there's a huge, vibrant city that are waiting to be explored. Thank you very much.
Thank you very much. I think a perfect summary of a perfect day, I have to say. We have question in any case, because it's a way now to open a bit this final discussion. We have a few minutes left, a few. One point, Pudong. There are some, let's say, extreme views that we should really activate inflammation in order to protect from neurodegeneration. There is a trial that is just now starting with exactly this type of intention to activate microglia in order then to try to block the degeneration in that will be initially in MS. Is a bit, let's say, in our mind, is a bit the opposite that what we have thought for a long time. What is your view about this?
Well, I wouldn't like my microglial cell to be activated because I think it's the risk of lighting fire within the brain. However, microglial cell and thus has the ability to clear the debris of of the products that are caused by neurodegeneration.